Dear students, today we shall be talking about structure and types of scales in fishes. Before proceeding, it is my request to you all to like and subscribe my channel BioLearnia. First of all, we should be knowing what scales are. When we try to define the scales, we say that the scales are small, thin, cornified, calcareous or bony plates that grow out of the skin of the fishes. So, the small, thin, cornified, calcareous, bony plates, whatever is present, because different fishes have different kinds of the exoskeleton or the scales modified as cornified, calcareous or bony plates that grow out of the skin of fishes. And this study of these very scales is called as squamatology. When we talk about the scales, they are dermal in origin. Dermal in origin. The genes which are involved in tooth and the hair development in mammals, they are also involved in scale development. So those very genes which are involved in the tooth and hair development in the case of mammals are also involved in scale development in these very lower vertebrates. As far as their arrangement is concerned, they are most often imbricated and thus overlap like shingles of the roof with their free margins directed toward the tail so as to minimize the friction of the water. Now what we see here that their arrangement is often imbricated. They look like as if we put on the tiles on the roof. We put on the tiles on the roof like this. They overlap one another. So just like the tiles on the roof. So what is happening here? This is the origin of the scale. This is the base of the scale. It is toward the head side and its free margin. It is toward the tail. Now this very arrangement is such that it helps to minimize the friction in the water, which is very much needed so as to reduce the energy required for the swimming. Scales and denticles of various kinds are the most complex derivatives of the integument, and they are derived from the mesenchymal cells. Now we have to keep in mind that they are derived from the mesenchymal cells of the dermis. Though their origin is dermal, and it is the mesenchymal cells from where they are derived. And these very scale represent an exoskeleton covering the skin in most of the fishes. Only few fishes are naked, having no scales in the body, such as electric, that we shall be dealing later on, electric rays, catfishes, cyclostomes. Though many books have written the cyclostomes, but they don't come under the series Spices. We call them as the Agnathostomata, but here in many of the books they consider them as the jawless fishes. That is why we often use these very cyclostome, or we also mention the cyclostomes in this very part as well. A few species, such as we do have the Polyodon, we do have the Accipenser, they exhibit intermediate condition. So whatever the modern condition is there and the most primitive condition, these very Polyodon and Accipenser, they exhibit the intermediate condition, having scales or plates only at some localized regions of the body. This is body, only some localized regions of the body. So they are present only on the localized regions of the body. So whereas in some the scales are present on the whole body, but in some they are present only in the localized regions, and that is in the case of Polyodon and Accipenser. Now we shall be talking about the types of the scales. Basically, the scales are divided into two types, that is Blackeyed scales and non-Blackeyed scales. When we talk about the Blackeyed scales, it is the characteristic feature of the elasmobranchs, that is cartilaginous fishes. And non-Blackeyed scales, as the scales of the teleostean fishes, the non-Blackeyed scales can be cosmide, these can be Ganite, these can be cyclide, and these can be Ctenide types. So basically, scales are divided into two, that is placoid scales and non-placoid scales. Non-placoid scales are further divided into cosmide, ganide, cyclide, or tinide types. Now let us look at this very chart. That is a classification and occurrence of the scales. This was put forth by the Wiechert 1970 and it was modified by Langler et al. In 1977. Now herein what we see that these very fish scales, they are divided into two, that is placoid and non-placoid scales. When we talk about the placoid scales, these are the characteristic feature of the elasmobranchi. And non-placoid scales, they are further divided into the cosmide, Ganite, and Bony ridge scales. When we talk about the cosmide scales, they are the characteristic features, or they were present in the extinct lobe fins and extant latimeria. That is lobe finned fishes, the extinct lobed fin fishes, whereas only the living fish, which represents the scales, which is cosmide type, and that is latimeria. We also call this very fish as the living fossil. And then we do have the Ganite scales and Bony ridge scales. Ganite scales, they are further divided into lepisostide and paleoniscoid. Paleoniscoid Ganite scales, they are present in the polypterus, whereas lepisostide, they are present in the lepisosteus. So Ganite scales, they are of two types, paleoniscoid, the characteristic feature of the polypterus, and lepisostide are which are present in the lepisosteus. Then the Bony ridge scales. Bony ridge scales are further divided into two, that is cyclide and tinide scales. The cyclide scales, they are found in the soft rayed teleos. And then we do have the tinide scale. It is found in the spiny rayed teleos, that we shall be elaborating just after a few moments. First of all, we shall be talking about the placoid scales. We also call them as the dermal denticles. Dermal denticles. They are found in the sharks. In structure, when we talk about the structure, placoid scales resemble just like the tooth. Though placoid scales are closely but do not overlap each other. I was talking about the scales in my first slide, wherein I told you that the scales are mostly present in an overlapping fashion. But in the case of these placoid scales, they do not overlap one another. And each scale consists of two parts. One part we call it as the rhomboidal basal plate. This is called as the rhomboidal basal plate. This is rhombic in shape, that is why it is called rhomboidal basal plate. In the dermis, it is embedded in dermis. And the flat end, this is trident, you can say spine, which is projecting outward through the epidermis. This very basal plate consists of the calcified tissue. So it has the longer one, which is called as the median spine, and these two lateral, are called as the lateral. The adjacent ones, we call them as the lateral spines. And according to Wiechert 1970, it is a bony plate which indicates the relic of the ancestral bony armor. It is anchored into the dermis by the connective tissue fibers. We call them as the Sharpley's fibers. So these are embedded in dermis, and they are very much anchored to the dermis by the connective tissue, which is called as a Sharpley's fiber. This is very important point, that these very placoid scales, they are embedded or anchored into dermis by the connective tissue fibers, which we call them as the Sharpley's fibers. Let us talk about the development in the placoid scales, that how development of the placoid scales takes place. The development begins with the multiplication of the dermal cells. Now, first of all, we should be well aware that it is only because of the multiplication of the dermal cells which accumulate at different points below the stratum germinativum. So just below the stratum germinativum, I'm just showing you, this is the Malpighian layer. This is also called the stratum germinativum. This is called stratum germinativum. So this very development begins with the multiplication of the dermal cells which accumulate at different points just below the stratum germinativum. So these very dermal cells, they form a papilla which grows outwards and pushing the Malpighian layer into strongly arched structure. Now what is happening here? That this very dermal papilla is formed, and it pushes this very Malpighian tubule to form an arch-like structure. The cells of these very Malpighian layer, they become columnar and glandular, forming the animal of the organ. So now what happens with this, that when it becomes arched, the cells of these very Malpighian layer, they become, number one, they become columnar, and they also become glandular, that is ability to form the glands. So they are now glandular as well as the columnar, and they form the animal like organ, animal. This very dermal papilla, when we talk about the dermal papilla, this very dermal papilla, which is just shown here, that this is the dermal papilla. This dermal papilla assumes the shape of the basal plate, and now this very dermal papilla, it assumes the shape of the basal plate now, and a spine. This very basal plate and then a spine. These very cells of the surface of the papilla form the distinct layer, and now they are called as the odontoblasts. So herein, now what is happening here? After this was the extreme I showed you, but actually what happens, that this is forming the animal organ now, and then it is forming the odontoblast. This is forming the odontoblast. These very dentine is formed, odontoblast is formed, and they secrete dentine toward the outer surface to form the papilla. The central cells of the papilla, they do not calcify and form the pulp. And then these are central cells of the papilla, which do not calcify, and they form the pulp. Herein the pulp is shown, which is not calcified. This very region is not calcified, and the spine of the scale is gradually pushed toward the surface, and before eruption, the cells of the animal organ called as the amyloblast, they secrete a layer of the vitrodentine. Now this is the vitrodentine which is secreted by the amyloblast. And they secrete a layer of the vitrodentine to form the cover over the spine. So the mesenchyme cells of the dermis surrounding the dermal papilla secrete a substance very much similar to that which is present in the teeth to form the basal plate of the scale. So basal plate is layer which is just similar to the cement of the teeth, and it is the basal plate of the scale, and thus the placoid scale has been regarded as of dual origin. So that is why we call it that it is of the dual origin, that it is partly dermal and partly epidermal. So when we talk about the placoid scales, we usually say that it is partly dermal and partly epidermal in origin. So now when we look completely over the section of the epidermis and the dermis in the case of the cartilaginous fishes which bear the placoid scales, we clearly see that this is a spine. The spine is projecting above the skin, while the basal plate, we do have this very basal plate which is actually made up of the cementing material. It is embedded in the dermis, and it is no longer supported. This very spine is no longer supported because it has animal It has the covering derived from the ectoderm, and its presence in the placoid scale is denied, and instead it has vitrodentine, that is the which is mesodermal in origin. So therefore placoid scale no longer enjoys the status of the double origin. And the one thing I was telling you about, that the basal plate, just near the basal plate we do have the certain connective fibers, these very connective fibers, these are the actually the Sharpley's fibers. So these are the connective fibers which anchor these very placoid scales to the dermis of the skin. Now the second category among the non-placoid scales, it is the cosmite scale. These scales are abundantly found in Sarcopterygii, Crossopterygii, and Dipnoi. So cosmites found in the primitive members of the Sarcopterygii, Crossopterygii, and Dipnoi. The only living member with the cosmite scale is Latimeria, which I told you earlier as well, that this very Latimeria is also called as a living fossil. And the cosmite scales are regarded as the precursor of the Ganite, Blackeyed, and the Bony scales of the modern teleos. So these very cosmite scales, they are regarded as the precursor of these very modern scales. So each cosmite scale consists of the 3 layers. Now what are these very 3 layers? The lowermost layer we call it as the isopedine. Then it has the vascular spongy, you can say, we also call it as the vascular layer, and then it has the cosmine. So isopedine, vascular layer, and then cosmine. These are the 3 layers which are present in the cosmite scales. When we talk about the isopedine, this is the inner, that is the basal layer. It consists of the layered bone. It consists of the layered bones. So it is pierced by channels of the blood vessels as well. What we see in them, that there are channels of the blood vessels as well. So these very isopedine, they are the basal layer and consisting of the layered bones. Then we do have the vascular layer. This is the middle one. This is the middle one. This is the middle one. And it contains numerous vascular spaces. That is why it got its term as the vascular layer. Then we do have the cosmine, the layer, the upper layer, that is the cosmine. This is the outer layer made of dentine and containing pulp cavities. So this layer is made up of dentine and contains the pulp cavities. Second kind of the scales are the Ganite scales. These are further divided into the, I was telling you about, they are further divided into the paleoniscoid and lepisostoid. So paleoniscoid Ganite scales, such scales are found in primitive and extinct actinopterygans, and on living chondrosteans. So the living chondrosteans do have the presence of these very Ganite scales, otherwise they are present in the extinct actinopterygans. And polypterus, that is the polypterus, the polypterus show this kind of the scales. What is the, it has further the isopedine layer, it is present. The spongy bone is absent here. In this case, what we see in the case of the Ganite scales, the vascular layer, but in this very case spongy bone is absent. The cosmine layer is reduced cosmine layer, and above the cosmine layer is multi-layered ganine is present in the case of the Ganite scales. And this, it is because of the ganine, it lends lustrous sheen, that is the shining. It is because of the ganine these scales do shine in these kind of the fishes. Then the second category we do have in the case of the Ganite scales, we call them as the lepisostoid Ganite scales, and such scales are present in the recent chondrosteans, and such as we do have the example in the form of the Lepisosteus. We call them as the gar fishes. So in this kind of the scales, what we see that it has the spongy bone and cosmine layers both absent. That is spongy bone, that is the vascular layer, and the cosmine layer, they both are absent in these very cases, but the ganine contains many tubules. So this is the ganine layer is present, and such scales are arranged closely together like the tiles on the floor in the diagonal rows. Now what we are seeing here, we are seeing the isopedine layer, ganine layer, and above it obviously epidermal layer is there and denticles are there, but here in this very case cosmine layer and the spongy bone, they are absent. Then now we shall be talking about the bony ridge scales. So these are of two kinds. We call them as the cyclide scales and we call them as the ctenoid scales. Cyclade scales, they are characteristics of the soft layered teleost and modern lobe-finned fishes. So cyclide scales, this is the characteristic feature of the soft red teleost and modern lobe-finned fishes. And the tenai means comb. So it looks like the comb, that is why we call it as the ctenoid scales. Ctenoid scales differ from the cyclide scale in having the comb-like teeth or tenai in its posterior margin. So these scales are composed of two layers. So only two layers are present, that is the isopedine layer and inner dense fibrous layer. So in this very case, in the case of bony ridge scales, you should be noticing and you should note that they are composed of two layers, that is isopedine layer and fibrous layer. In this very bony ridge scales, the scales are arranged in an overlapping fashion like the tiles on the roof of a house, and they show different kinds of the features. Now what are those very features? Let us have a look. The number one, this is the focus. This is the focus. This is the central zone or the nucleus of cyclide and ctenoid scale and is the first part to develop. So whenever the development of the scale starts, it is the focus which forms. The focus is formed, and in some scales focus may be away from the center, but in some cases it is in the center. And secondly, we do have the circuli. Circuli, what are these very circuli? The lines around the which cover this very focus in circle, we call them as the circuli, and these are the concentric series of these are the series of the concentric bony ridges around the focus. We also call them as the lines of the growth or growth rings. And thirdly, we do have the radii. We do have the radii here. These are the grooves that radiate from the focus toward the margin of the scales. So rightly from the focus toward the margin of the scales, some lines radiate and they form the groove. We call them as the radii. Then one important thing is there that many times we see the annuli, that these are called the annual ear growth marks. So that is a different thing, that how these very growth marks are formed. But many times what we see that these very growth rings, they are concentrated at certain places and they form the yearly growth rings. Then it has the anterior margin, this very margin, anterior margin, and these lie in the embedded part of the scale. In the ctenoid scales, the posterior margin is readily recognized. In the case of ctenoid scales, recognizable. Now how? Firstly, the ctenoid scale, we see that when it has the comb-like teeth or tenai, these are the ctenoid scales. If it is the round, we call it as the cyclide scale. So cyclide scales are recognized by the presence of indistinct or eroded bony ridges, that is the circuli. Whereas if it is the comb shaped, we call them as the ctenoid scales. So both cyclide and the ctenoid scales may occur in the same fish. Many a times there are certain fishes which have both these scales on the same you can say body. For example, Pleuronectus is a fish in which the scales are present on the pigmented dorsal surface Ctenide scales are present on the pigmented dorsal surface, while the cyclide scales are present on the lower side. So on the lower side, the cyclide scales are present, whereas on the pigmented dorsal side, the ctenide scales are present. The origin of these very scales. How these very scales originated? There are different views. The scales originate as the thin disc-like bones in the deeper layer of the skin. These are remnants of the bony skin of ostracoderms and placoderms. These are remnants of the bony skin of ostracoderms and placoderms. When we talk about the teleos scales, that is the cyclide or ctenide scales, they are derivatives of the Ganite scales, but the ganine and the cosmine layer have been lost in them, and thus teleos scales are thin, soft, and flexible dermal plates. And the Blackeyed scales are nothing but the dermal denticles. Their entire origin is mesodermal alone. So what are the derivatives of these very scales? That in some fishes these very scales have the certain derivatives. For example, in the Blackeyed scales, when we talk about the Blackeyed scales, jaw teeth in the case of sharks, they are known to be as a derivative of the Blackeyed scales. These saw teeth of the pristis, they are also known to be the derivative of the Blackeyed scales. Then the spine in the case of chimera, the or the dogfish, we do have the spine in the case of the dogfish, squalus, and chimera. They are also known to be as the derivative of these very scales. Then we do have the sting, which is present on the tail side. In the case of the trigon, that is also known to be the derivative of the Blackeyed. There are certain derivatives of the non-Blackeyed scales as well, such as soft rays or lepidotrichia. That is, those very soft rays which are derived from the proximal radials, toward the margins. We call them as the lepidotrichia. Then dermal bones in certain cases, or the bony cuirass of the seahorse, bony cuirass in the case of seahorse, the cowring, and in the case of ostracion, that is the boxfish, that is also known to be as the derivative of the non-Blackeyed scales. Then the spines, bony rings of the pipefish, bony rings in the case of pipefish, that is also known to be as a derivative of the non-Blackeyed scales. And the spines of the porcupine fish are the diodon, that is also known to be as the derivatives of the non-Blackeyed scales. Certain modifications and variations of the scales. That is, scales are variously modified and there are variations in the case of scales. So there are certain fishes which are without scales, and they have evolved certain other alternatives for the production, and they provide such as tough leathery skin or the bony plates instead of the scales. So many eels, they are scaleless. Though some species are covered with tiny smooth cyclide scales, but most of the eels, they are known to be scaleless. And in the eels, the scales, that is cyclide scales, they are minute and deeply embedded in dermis. And we do have the catfishes. They lack scales, and the several families have body armor in the form of the dermal plates or some sort of the scutes. So in certain, the scales, they are in the form of the body armor, in the form of dermal plates or some scutes. And the mandarin fish, this is the mandarin fish. These very fish lack scales and have the layer of smelly and bitter slime, which blocks out disease and probably discourages predators, and implying their bright coloration is also opposematic. So instead of the scales, they have developed certain other mechanisms in order to protect themselves. Then we do have the anglerfish. They have the loose skin. Here in this very case, in the case of angler skin, they have very loose skin that is covered with the forked dermal prickles or tubercles, and they do not have the regular scales. They rely mostly on the camouflage to avoid the attention of the predators. while their loose skin makes it difficult for the predators to grab them. So that is how they have modified themselves, by not having the scales but having the loose skin. And many groups of the bony fishes, including these very pipe fishes, or we do have the seahorses, box fishes, poachers, and several families of the sticklebacks. They have developed external bony plates, structurally resembling Blackeyed scales as the protective armor against the predators. So in all these very cases, that is the bony fishes, including the pipe fish, we do have the seahorses, we do have box fishes, we do have the poachers, then we do have the sticklebacks. They have developed the external bony plates, which structurally resemble Blackeyed scales as the protective bony armor. And the seahorses, when we talk about these very seahorses, they lack scales. They have thin skin that is stretched over a bony plate, which is stretched over a bony plate armor, arranged in rings through the length of their bodies. That is, their bony armor is ring shaped and stretched over their body. And when we talk about this very box fish, the plates fuse together to form the rigid shell or exoskeleton, which encloses the entire body. So these plates are not modified scales, but the skin that has been ossified. So here in this very case, the skin is ossified. And because of this heavy armor, box fish are limited to slow movements, and it is because of this very not because of these very this very kind of the exoskeleton which is rigid, and it is because of this the in the case of the heavy armor, in the case of the box fish, their movement is very slow. Some fishes like hockey, this is the hockey, and the swordfish, that is a very known fish. They are born with scales. When they are born with scales. That is, when they come out of the egg, or come out, or they are born, they are born with scales, but later on they shed their scales as they grow. So there is one fish known as the filefish. They have the rough non-overlapping scales with spikes. This is the filefish, having the rough non-overlapping scales with small spikes, which is that is why they are called as the filefish. Just like the spikes of the file, resembling to that of the file, that is why they are called the filefish. And some filefish appear scaleless because their scales are so small. Then they do have the prominent scaling that appears in the case of tuna. So when we talk about the tuna, a prominent scaling appears on tuna only along the lateral line, and in the corselet, a protective band of thickened and enlarged scales in the shoulder region are present. And over most of their body, tuna have scales so small that to casual inspection they seem scaleless. So they have the scales present, but when we see casually, we see them casually, it appears that they are scaleless, but minute scales they are present over most of the body. They have the scales, but very small and minute, hardly to be noticed. Or let us have a quick revision what we have read in these very modifications. In the globefish, that is Tetrodon, and porcupine fish Diodon, scales develop into protective spines. So in the case of Tetrodon and Diodon, the scales are modified as the protective spines. So some fishes are totally scaleless or naked, such as torpedo, electric ray, or catfishes. Then we do have the Syncheiropus splendidus, which we call them as the mandarin fish. They lack scales and protect themselves with a layer of the smelly bitter slime. In the trunkfish, that is Ostrachion, the scales are form a complex bony box. That is why we call them as the box fishes. So in the case of sturgeon, that is Accipenser, stout bony plates or scutes, they form the bony armor. In the case of hippocampus, that is seahorse, or Syngnathus, that is pipefish, the scales become fused to form the protective bony ring around the body. And the shark teeth and the sting of the stingray, they are regarded as the modified scales. Now let us quickly know about the uses of the scales. That what is the importance of the scale in the in case of the fishes. How the scales are useful. The scales are useful for the classification and identification of the fishes. That is, they are used in the taxonomy of the fishes. The scale count along the lateral line, they are used for the cyprinids to know about the meristic characters and thus help in the taxonomy. The scales are useful for the determination of the age in the case of the fish. So there are seasonal fluctuations in the growth. In summer, when the food supplies are abundant, the fish grows rapidly and so do its scales. In winter, the growth slows down due to the temperature dependent metabolic processes. The wide lines, that is, which are separated by the lines of the growth, they represent the growth, and the closed lines, they represent the slow growth. And a series of wide lines and a series of the closed lines in an area on a scale, they correspond to a year in the life of the fish, and thus from this we can know that what is the age of the fish. And thirdly, the scales are useful in the estimation of the number of the spawnings as well. So the spawning marks may be seen on the scales of the salmon, which is a migratory fish, and during the anadromous journey, the fish do not take food. Due to the calcium deficiency during the fasting period, erosion of the scale margin occurs and leaving the spawning marks. And besides, it is obvious to say that in the case of the fishes, they are protective in nature. They form the exoskeleton of the fish. They protect the fish from the different, you can say, many times from the different diseases and many times from the different predators as well. There is one term called as the lapidophagy. There is one term called lapidophagy. When we talk about the lapidophagy, it is an ancient Greek word for scale eating. That is, those very organisms which are specialized in feeding, whose specialized feeding behavior in the fish involves the eating of the scales of the other fish. We call such a process of the feeding as the lapidophagy. So those very organisms which feed on the scales of the fish or the scales of the other fish, we call them as this very term is called as the lapidophagy. So this was all about the scales, their types, modifications and variations, as well as the uses of the scales. Thank you for watching this video, and hope to like and subscribe my channel by pressing the bell icon. Thank you very much.